This is a production of Cornell University.
Thank you. Thank you for the introduction. I'm honored here to be present today and present my talk in front of all of you. Particularly, I want to thank the Graduate Student Association for organizing my visit and being so generously accommodated of my travel plans, which have changed drastically since last night, which is why I have to leave early, which is why I will try not to talk too fast. But hopefully, I'll have you interested in my work in incipient soils. So my story of soil starts with this pretty nice forest picture on the left-hand side. This is from a site, one of my field sites in Ohio, where I worked. This was near Wooster, Ohio. And when we think about soil, that is what we generally think about, right? It's a well-developed system. There's lots of plants growing in there. It's lush and happy and all of that. But there is another system of soils, which is on the right-hand side. This is a landform, a lava landform in Hawaii. And you see that house up there, they actually sell plots there, which are super cheap. You can put up an Airbnb there because the land just, new lava forms and it all disappears and stuff. So if you want property, that's a good place, but high risk. So that is an incipient landform. That is one of the landforms from where soil starts developing. It's a basalt landform, right? And that is where my system, that is what I study. But before we go there, so this entire process of an incipient soil to a developed soil is a long process. Just even 1 inch of topsoil about takes 500 to 1,000 years to form an inch of topsoil, right? And all our focus, most of our focus is at this end. And it doesn't matter which end you're in. I want to bring to your attention that microbes play a crucial role no matter if you're in an incipient soil, in a developed soil, in a young soil. And of course, microbes are, we know they're less than 0.1 millimeters. They are these group of organisms that are present in soil. And really, no matter where they are, again, they participate in organic matter breakdown. They participate in nutrients recycling. They exude substances which bind the soil together. And I don't have to explain why microbes are important in soil to this group. And I also don't have to spend a lot of time explaining the diversity, right? We are grappling with this issue that soil is highly microbially diverse and we don't know what's in there. We don't have an exact count of who is there and we don't know what they're exactly doing. And I read somewhere that an acre of soil has bacteria that weighs equal to that of 2 cows. I don't know how that was estimated, calculated. Not sure about that, but there are numbers that range from you have 1 billion microbes to more than 1 million species. And we have accepted that the numbers are large. We don't have a concrete number, but that's how we operate, right? So obviously, again, it's highly complex. The system is highly complex. For those of us who work in soil microbiology, we know that this new view of tree of life that came out, this is eukaryotes, this is where we fall. And all the red dots that you see are representatives that have been confirmed from their sequences, but we don't have cultured isolates. We don't have physical presence or confirmation of these groups of bacteria. All the red dots that you see, and especially this one is called the candidate phyla radiation. There isn't a single cultured representative that's present, so highly complex. And with that complexity comes the fact that if you will imagine, each microbe likes its own living room, and it wants its living room to be super comfortable, right? So it has a unique pH requirement, unique oxygen requirement or not, moisture content or not, right? So when you have this high diversity, so many numbers, with each requiring its own sort of living room to speak of, we are dealing with a very complex system. And as somebody said once in a conference that I was, they said, well, you have chosen to study the most complex system on Earth. So that made me feel happy. So good for us. The second challenge that happens is these processes occur at multiple scales, right? So for example, as Janani pointed out, I was studying methanotrophic bacterial population in agricultural soils. With this understanding that if methane that eats methane gas, so if you have more of methanotrophs, they will eat more of methane, there will be less emission from the soil, right? So that is a greenhouse gas emission aspect to the study I was doing. But that really functioned at this scale, right? We want to find out what microbes are doing globally to our environment. We answer those questions at the landscape scale. We go out to the field, we set up our study system, we sample. But really the processes that I mentioned, which makes microbes so important in soil, occurs at these really smaller scales here, ranging from meter to nanometer scale, right? So how do you match this and how do you map back or map down? And this is the second challenge that exists in soil microbial ecology. And some of us, as we know, the Earth Microbiome Project is making many efforts to categorize microbiome of the Earth, and not just restricted to soils, but plants, human body, animal gut, and so on. So they came out with this seminal paper last year. If you don't read any other paper in soil microbiome or the Earth microbiome, you should go read this one. And then this one, which kind of said, okay, too many stuff in the soil. Let's narrow down and find out what's the dominant ones and how they're characterized over time and space. Again, if you need no other paper, you want to understand soil microbiology as it exists today with the new technologies that we have, this one is a good paper to refer back to. And this came a month back. Soil is good, but most of these studies, and I would almost say 90% of these studies, are done in developed soils. Again, they're not looking at incipient soils. They're not looking at marginal soils. And by marginal soils, I would say soils that are degraded, soils that are incipient, soils that have been left fallow. Not all those systems actually show up in these kind of studies, which is what makes my work super interesting and where I find myself. So I'm a postdoctoral researcher at Biosphere 2, University of Arizona. And we have this beautiful setup here where this is the rainforest. We actually have habitats in there. So this is the rainforest. Somewhere here is the ocean and the desert habitat. And these three things that you see here, these three basically, they consist the Landscape Evolution Observatory. The Landscape Evolution Observatory is basically a tennis court size artificial landscape. And there are three of them. So we need replicates. So we decided one landscape is not enough. We are going to build three of them. It's highly climate controlled, so we can regulate the temperature except for freezing temperatures. We can regulate wind speed. We cannot regulate solar radiation. That's something we have to accept we can't. But it's highly controlled, and it's basically studying landscape evolution from the perspective of hydrology, geochemistry, and microbiology. So what happens at that starting time point, and how the landscape evolves over time, how these three things actually evolve together in the landscape, and can we identify those tipping points? Can we identify the actual points where the shifts occur dramatically in a landscape? This is a highly climate controlled environment with almost within the soil system, there are 1,800 sensors and samplers that are recording data real time. These are very large data sets. These are available to the public if they want to work with it. It primarily started out as a hydrology project, but we have geochemists and microbiologists working alongside the hydrologists on this. The landscapes are filled with crushed basaltic tephra. These are super low in carbon and really, really, really low in microbial DNA. So that makes my work even more exciting because I work with a system that has pretty much nothing to start with. So yeah, the sensors record pore water chemistry. The samplers record pore water chemistry. The sensors record carbon dioxide measurements, soil moisture potential. We have pH measurements, CO2 measurements. There are above ground sensors for for radiation, for wind speed, for temperature. So really, we can control everything that's happening in here. And what you see here, the masts, these are the sprinkler systems. So we can irrigate as much as we want with the rates that we want. So water is not a limitation, even though we live in a desert. And it sounds ironic, but inside Leo, water is not a limitation for us. These are 1 meter deep landscapes and 30-meter long and 11 meters wide. They have a sloping angle. So basically, if all water collects down here and flows out from all the ends. And these are two of the sort of the Pangle 2015 is the first snapshot of the Landscape Evolution Observatory. And the next one is a book chapter where we have introduced the advanced instrumentations that we have in the system over time that we developed. A lot of those are available in the second reference. So before I forget, it takes a village to do science, and especially in a system like Landscape Evolution Observatory, these are multiple collaborations. I have 3 postdoc advisors, which a lot of people go like, wow, but I find it really exciting. So I have a hydrologist, Dr. Peter Troke, John Cherover, who is a geochemist, and Dr. Reina Mayer, who's a microbiologist. And then I also collaborate with PNNL and EMSL, the national labs, for quite a bit of our analytical measurements that we do. And then there's Laura Meredith, who's a new professor at UFA, and she works with trace gases, and we're trying to link microbiome of trace gases, basically, in this system. And then there's Still, who's another postdoc, he's a hydrologist. And then Yari and Antonio, they're the graduate students that I work with. And really, this work is not possible without so many of us involved in this. So it's a really cool collaborative place to work at. So what am I working on, right? My main question is how microorganisms establish, evolve, impact and respond in an incipient soil system. And I'm asking that question in the perspective of these three boxes that you see here, with this understanding that these are all connected with each other. They are not isolated, right? So if your hydrology changes in some way in an incipient system, your microbiology and geochemistry are going to change, which are then going to feed back hydrology and geochemistry in turn. With that understanding, but not exactly knowing how they feedback, we don't know those feedbacks yet exactly. That's where my work comes in, right? So I am going to present two study systems and hopefully do a good job of not rushing through very fast. So this one system we work on is the mini Leo, and the second system is obviously Leo that I'll talk about. So mini Leo is a guinea pig for us. We can't, sorry, I didn't mean to put the laser on your eye if it went, I'm sorry. The Leo hill slopes, we cannot core on them very frequently. We cannot disrupt the landscape as much. Also, a very cool trivia fact, we cannot step foot on the landscape. So we harness ourselves to the ceiling and take soil cores basically, because we want to keep the soil undisturbed. Having said that, we work as a first pass, everything we do is done on mini Leo. And mini Leo is a scaled down version of Leo. It's still 1 meter deep, so we have maintained the depth profile. It's filled with that same basaltic tephra. It's still instrumented a lot. Here you see on the right hand side are these pore water discharge tubes that go out. On the left hand side, there's the instrumentation box that again records, the sensors are recording time real time, right? And there's a sprinkler system here. So pretty much everything is similar except the size. So when I joined UFA, this was my first project. I had to take apart this entire thing and study its microbiology. So I had to look at the community composition of mini Leo. Then I took that step forward and I wanted to understand ecologically, how were the microbial communities assembling in Leo, right? How were they talking to each other? How were they congregating at one place or not? I don't know, I didn't, so we had only our hypothesis. And then can we, because this is such an incipient system, is there carbon in the system? Can we identify that? And what kind of carbon metabolisms are happening in the system? So I'll present us a brief snapshot of the results. This is not all the results that we have, but hopefully it'll drive home the point. So obviously this is what we want to study. This is again mini Leo in its grandeur. What we did was rained on it for 2 years with this controlled precipitation, which of almost 15 meters of precipitation. And the hypothesis that we had is because of the precipitation regime, there would be a stratification in environmental conditions in this mini Leo. So even though this is a homogeneous basalt soil system, the stratification as a result of redox conditions, water potentials, is going to give rise to a heterogeneous microbial community. And then the objective was that, can we capture this heterogeneity in the first place? Do we see the heterogeneity and can we capture it? And then how is that heterogeneity happening? And this small scale that I point out, I'll come to it in the next slide. This was a very intensive sampling procedure. We basically broke down mini Leo into these voxels, so like cubes, and we sampled, we took one voxel from the surface, the next one, the next, and next, and next, 10 in a row. There were 4 rows. We had 40 samples from the surface, and then again 10 centimeters below 40 samples, then next 40, and next 40, and next 40, so on. So we ended up with more than 350 samples. And that each voxel that you see, we collected from that each voxel, which is about this size, we collected a microbial core, a bulk density core, and then hydraulic conductivity cores. And whatever was left behind was for geochemical analysis. And we had to make sure that our sampling was, we felt like archaeologists, basically. We were using all the tools that archaeologists use to dig up soil. And this is a real-time picture of how one voxel looked like when all of us went in and did what we had to do. And my role was to, basically get the community understanding of microbes using sequence data, and then work on some assembly models. The the geochemistry team went and analyzed carbon, nitrogen, pH, elemental composition. The hydrology team did these bunch of calculations and also ran some models to basically understand the flow path regimes of the system. So very detailed hydrological analysis that went on as well. So what did we find? The question of, do we find heterogeneity? Yes, we found heterogeneity. And a really quick snapshot is all these that you see are the names of microbial phyla, right? And the different colors represent different kinds, and this is the relative abundance. So if you compare that to the parent material and the irrigation water, straight away there is a difference. Even if you don't understand microbiology, there is a difference in the numbers of these groups that we see, right? Which means there are more in Menilio than there was in the parent material or the irrigation water. Not only is there more, but there are of different abundances. They're not equal. Some is like, this one is super high, this one is like, well, not so much cyanobacteria, which we were hoping we would be able to see because that is a photoautotroph. It fixes carbon. Surprisingly, we did not find a lot of cyanobacteria in there. So something in the soil system is unique. So yes, there is heterogeneous community composition. Then the next question we wanted to ask is, how is this composition distributed, right? So we looked at the depth profiles and the length profiles. So the way to read this plot is every dot here is a sample, and the closer they are means they're similar to each other in their community compositions. So microbes in this dot and this dot are more similar to each other than microbes in this dot and this dot, for example. So the surface samples really stood out really uniquely. And then there was a depth gradient in the community composition, which sort of diffused as we went lower and lower and lower. So something unique is happening on the surface here of Menilio. And this region, probably the water table is about at this 30-40 centimeter zone, and that probably is driving this cluster where they constantly see water all the time for most part of the experiment. So we see heterogeneity, we see depth-dependent heterogeneity. Then the next question that I wanted to ask is, can we understand how these communities assemble together? Which basically means in ecology, the degree to which a community is similar or different due to ecological processes. And they could be selection processes, they could be dispersal processes. So what that essentially means is, for example, if you imagine this as a cross-section of Menilio, right? You're just looking at it from one cross-section. What the community assembly asks is, how similar or different are your communities in the green box as compared to the purple, as compared to the blue, as compared to the lilac, I don't know if that's lilac, or as compared to even this one down here. And if they are similar, why? If they are different, why? That's what, in broad, microbial community assembly means. And the way those differences occur is due to deterministic or stochastic processes. So again, imagine a mesh, right? And you have these number of different kinds of bacterial or microbial cells. Deterministic means there is some sort of selection process that's going on, which essentially allows only the purple ones to pass through the mesh. On the other spectrum, you have high stochasticity, and nobody really knows. It's chaotic and everything passes through. And in somewhere in the middle, you have a moderate stochasticity where some cells pass through and some cells do not, right? And that broadly defines whether your community is deterministic. So if it's deterministic, then for us, we can predict. If a community selection is always happening deterministically, we can predict what it will look like in a year's time or 5 years' time or whatever. So that gives us a lot of control in terms of understanding the community. If it's stochastic, that helps us as well. We just know we cannot predict. We still don't have the controls or factors which might define how a community is structured. So the results that I found, so first of all, to read this plot, this is the surface of Menilio, right? And this is the depth profile. And this is basically the toe slope. So all the water is discharging out from this, right? The more blue it is, the higher influence. The more red it is, the lower influence. Okay. So what we saw is on the surface, there was a lot of blue. And it was deterministic. Some sort of variable selection was occurring, which we think is abiotic. And that was giving rise to this very distinct biotic community on the surface. And also at this central, about 40 centimeters, 40 centimeters depth, we also found a high presence of blue influence, which again was something here was also deriving what the communities that we saw. This was surprising to us because we had thought that the system is so chaotic that we would just see stochasticity. We would not see deterministic properties in any way. But stochasticity, which is dispersal limitation, which is, do you expect that communities just exchange randomly? So remember the cell picture. So within the green cell and the purple cell, communities are randomly moving. Or there's random birth death events in those green versus purple versus orange cells. And none of those happened. So stochastically, we didn't see any evidence, which is super interesting because we are studying a homogeneous system, right? Has nothing in it. As a microbial ecologist, extracting DNA from these soils is really challenging. And it takes us 3 times the normal time that it would take for any of us to go sample the soil here, like to extract DNA from these soils. So you're spending a lot of effort. And we could capture these patterns only because we did such an exhaustive sampling procedure. We went and sampled block by block and chunk by chunk. We wouldn't have been able to run these models or get these results if our sampling strategy was not this extensive. So moving on from there, there's a bit of geochemistry work, biogeochemistry work that's going on, keeping with the storyline of low carbon systems. I'm working with EMCEL, which is the national lab, part of the national lab at Richland, and using this FTICR method, which is basically a mass spec method, where you take your soil samples and you have 3 set of extractions on them. So you extract with water, with methanol and chloroform, and you get organic compounds. You get organic compounds, then you can know what their hydrogen to carbon and oxygen to carbon ratios are. So the way these plots are read is each dot is a unique organic compound, and the intensity tells you more or less of it. So if you look at one sample from the surface of Menilio and compare it to a sample that was 80 centimeters below the surface, you can see that the dots differ in certain regions, and the intensities also differ. So in a system where the carbon is so low, a technique like this mass spec, which is a very ultra high resolution technique, can tell you what the system looks like. So if you don't have the ability to go and do microbial DNA extractions, you can basically take a genome inspired approach. How? You have compounds, you know which pathways produce it microbially, because plants, these systems don't have plants, right? So how are they in the system? They are probably they're micromediated. So you know which pathways they are, you know which enzymes produce those pathways, then you can backtrack and figure out which genes produce those enzymes, and basically develop a map of the microbial metabolisms that are going on, and hence derive the microbial community presence or absence in your soil system. So even if you don't have a lot of microbial DNA, these techniques are really cool to work with. Also, EMCEL has user proposals where you can submit proposals, and it's a user facility. They don't give you money, but they take your samples and they analyze it for you. They have something called the rapid access proposal, which is, I submitted my samples and it was like 30 samples and they worked it out for me. So it wasn't even a full-fledged proposal. So they have very good resources that we can go and sort of work with them to use. So the conclusions from the Menilio work really is we did find heterogeneity, which was primarily depth dependent. And that depth dependent heterogeneity was probably a cause of the abiotic conditions that were present in the soil. And the community assembly highlights, deterministically or stochastically, we can eventually map back to these organic compound characterizations in the biogeochemistry that is happening at each step. Even though we may not have redox fluxes, we not have exact measurements of those parameters, we can extrapolate. This is a very strong extrapolation method. And then incipient soils, they're suitable for understanding community assembly processes, because we always keep saying soils are highly microbially complex, we don't understand them enough. So my point is start from a simple system and then try to understand the complexity.
This has been a production of Cornell University, on the web at cornell.edu.